Hello, everyone. Thank you for joining me today for this presentation that I give to support the great endeavor of my good friend, Mr. Hugh Choi, an honorable man. Today, I wish to talk about two products I exclusively designed for Mr. Hugh Choi, Zinc-Zetabiome and Zeta-Roche B12. Before we start, please allow me to introduce myself. I obtained my MD in 1980. I was born in Firenze, Italy in 1956, so I was 24 when I obtained my MD. I have also a PhD in molecular biology and a specialization in diagnostic radiology. As a young postdoc, I worked at Burroughs Wellcome in the mid-80s in North Carolina, USA, and later as a visiting scientist at the National Cancer Institute of the NIH, US government, from 1987. Later on, I was appointed professor of molecular biology at the University of Firenze, Italy, in 1992, where I worked until 2014. I am the founder of Silver Spring, a company located in Switzerland that is specialized in research and development of innovative approaches and products, among which those that I designed exclusively for Mr. Hugh Choi. Over the course of about 40 years of career, I published more than 200 scientific articles with many co-authors, and among those I'd like to remember eminent scientists such as Eduardo Lapetina, Stu Aronson, Jackie Pierce, Peter Duesberg, Henry Bauer, Jeff Brestreet, Dietrich Klinghardt. One of my articles was sponsored by a Nobel laureate, Sir John Vane. According to metrics, I am in the top 2% of all scientific authors, and my articles are among the most viewed and downloaded. For example, this is Loop. That is one of the most reputable database for science. And as you can see in the benchmark, I am in the top 2%. My more than 200 publications have been viewed more than 36,000 times and almost 4,000 downloads. If you wish, you can go to my Google Scholar page. Just look for Marco Ruggiero on Google Scholar, and you can see all the metrics in the number of citations. My articles have been cited by other authors almost 5,000 times, the H-index, the I-10 index, and of course you can read all those 200 papers if you are interested. Here I wish to clarify my business and personal relationship with Mr. Hugh Choi and with his company, Danabaiologics. In the past 3 years, I have had the honor and privilege to collaborate with Mr. Hugh Choi, very good friend, an enlightened entrepreneur possessing unique talent and vision. I am proud to declare my esteem and admiration for Mr. Hugh Choi, and I deeply cherish the friendship and business relationship that binds us like a pact of steel. The fruits of this collaboration are two novel, original, unique, innovative, state-of-the-art products that I designed exclusively for Mr. Hugh Choi. One of these products, Zinc-Zetabiome, this one here, has a powerful immune-supporting, anti-cancer, anti-inflammatory properties. The other, Zeta-Roche B12, has powerful brain-supporting properties that prevent and treat Alzheimer's disease, dementia, neurodegenerative disorders, cognitive deficits, and help enhancing consciousness. Zinc-Zetabiome and Zeta-Roche B12 are the products of more than 40 years of research performed at the highest level as evidenced by my scientific achievements, and I'm proud to declare that these two products represent the apex of my scientific career. They are, without any shadow of doubt, the most innovative, revolutionary, exciting, and efficient products that I have ever designed or developed. Since science is an ongoing pursuit, and novel discoveries appear every day, the molecular design of these products is developed in such a way that they can be constantly updated and further improved based on our own research as well as on the general progress of scientific knowledge. I shall be honored if Mr. Yuk Choi will help develop other products, and I take this opportunity to declare that I'm fully available to design and develop other innovative products for him. It is a privilege to work with such an astounding person as Mr. Choi. Likewise, my greatest desire is to have Mr. Choi participate in my ongoing and future research. I am fully convinced that his contribution will be determinant. Joining our forces will lead to results that have the potential to change the entire world of medicine. My current research interests are in neuroscience, science of consciousness, immunotherapy, autism, and oncology. I am convinced that with the collaboration of Mr. Choi, we can achieve formidable results in all these areas. Zinc Zetabiome and Zetaroche B12 have been designed having in mind manufacturing efficiency, cost, and scalability. The intellectual property, IP, of the two products is currently under our full control, and the products are manufactured in a state-of-the-art FDA, Swiss, and Demeter certified facility in northern Switzerland. Once Mr. Choi will have acquired the full IP, the products can be easily manufactured in any part of the world, either entirely in-house or he can decide to externalize part or the entire production. The products and the manufacturing of the products is extremely flexible. The manufacturing process that is an integral part of the IP thoroughly describes step by step how to manufacture the products in the most efficient and cost-effective manner. And all this is written in the description of the IP. Ownership of the IP guarantees unrestricted, absolute power on all and every aspect related to the two products, from manufacturing to patenting, all over the world, marketing and sales. As clearly stated in detail in the signed IP transfer agreement, I am determined to provide full assistance and support for Mr. Choi in all aspects related to manufacturing and marketing the products. Since our Swiss company has extensive expertise in technology transfer, Switzerland is renowned all over the world for this. I am fully confident that manufacturing the products in the facility selected by Mr. Choi will occur smoothly with no problems at all. Zinc Zetabiome and Zetaroche B12 were designed on the basis of successful advanced research in the field of microbiome medicine, where I have been the first and the sole developer of a probiotic that has more than 400 different microbial strains, as they were identified by DNA analysis. The results of this exceptional feat were reported in 2 scientific articles published in prestigious peer-reviewed journals. This is the first one that was published in the Madrid Journal of Immunology in 2019, where I describe a number of strains and plasmids that were observed in the probiotic that I had developed. And this other article was published 1 year later in 2020, where I describe also the phage composition. Phages are the so-called friendly viruses that help us with the immune system, help us with cancer and bacterial infections. And in this article that was published in the Journal of Neurology and Stroke, I describe all the phages and their actions as they were observed in the probiotic that I had developed. Zinc Zetabiome and Zetaroche B12 draw inspiration from this research, and their development exploits the results described in these 2 articles that I have shown before. It should be noticed, however, that they are even better, because Zinc Zetabiome and Zetaroche B12 represent a much advanced concepts in comparison to those described in those 2 papers. They are even more innovative than those described in the articles that I mentioned before. The science behind the products that I have designed and developed exclusively for Mr. Choice has been presented at an international conference and has been the object of a scientific article published in the prestigious Journal of Neurology and Stroke. About 1 year ago, in Rome, Italy, there was this international conference on Alzheimer's disease and dementia, and I presented the results, and they were acclaimed by all the audience. And this is the article published in the Journal of Neurology and Stroke. Here, I describe how these products have the ability to optimize the function of microtubules and tubulin in the immune system and in the brain as they relate to the Kor theory of consciousness. This article is available in the public domain. You can download it for free if you like, and if you read it, you will find all the details about these products and the way they work at the molecular and quantum level. And I will talk about this later. Let me spend a few words describing what this Kor theory of consciousness is, because it's a revolutionary theory of consciousness, the most credible, the most reputable theory of consciousness that we have today. Kor stands for Orchestrated Objective Reduction. And this theory postulates that consciousness arises from quantum computations in microtubules inside brain and neurons. It was proposed about 20 years ago by a professor working at the Arizona State University, Professor Stu Ameroff, and a Nobel laureate, Sir Roger Penrose. And I had the privilege to attend a conference where I presented my results together with these two eminent scientists. And let me tell you that in about 1 week, I shall go to another conference where these two eminent scientists will attend, and I will present my results as well. Now, this theory is so important because it leads to new ways to diagnose and treat neurological diseases as well as disorders of consciousness. For example, it could lead to the development of new approaches that target microtubules and tubulin. And that is exactly what the two products I have designed for Mr. Yuk Choi do. In the article on the Journal of Neurology and Stroke, a novel approach targeting microtubules and tubulin is described. This approach is based on novel compounds deriving from the self-assembly of two types of information-containing aperiodic crystals. Again, all this is described in detail in the article. You can read it in detail. These are the compounds that I designed and developed for Mr. Choi. These compounds are able to support and optimize the function of microtubules and tubulin in the context of our core, as well as in the context of their role in the immune system. These formulas, that are these compounds, have the potential to prevent and treat a number of conditions, ranging from neurological diseases to immune deficiencies, cancer, and diseases caused by viruses. The quantum computation described by Kor are based on the phenomenon of quantum entanglement. This is a very strange phenomenon. Even Albert Einstein himself had difficulty understanding this. He called this a spooky action at a distance. There are several ways today that we can use to describe such an intriguing phenomenon. This is a very simple example. Imagine two coins, incredibly tiny and far apart. You flip them both, they can be head or tails, but not decided yet. You have not looked at them. When you look at one coin, the other instantly becomes the same, like magic. And that's kind of like quantum entanglement. Two particles, two atoms, even two bacteria are linked in a special way. So whatever happens to one instantly affects the other, no matter how far apart they are. It's a spooky, but it's a real physics phenomenon that now has been known for decades, has been demonstrated in all possible ways. And the products I've designed for Mr. Choi, they work on a quantum level. Quantum entanglement, as a matter of fact, is not limited to the realm of physics. Quantum phenomena, they happen in the biological world. There are 6 major biological phenomena that have been identified as influenced by quantum effects, enzyme catalysis, sensory processing, transference, consciousness, immune function, and information encoding. I personally worked on this field, and I contributed to quantum medicine with some notable scientific articles, among which I like to show these two, published both in 2018. One is entitled How Quantum Biology Can Eradicate Heart Diseases and was published in the Open Journal of Cardiology Heart Disease. The other one is entitled On the Impact of Quantum Biology and Relativistic Time Dilation, more about this later, in Autism. This was published in the Ames Journal of Molecular Science. As described in the article published in the Journal of Neurology and Stroke, the products that I have designed and developed for Mr. Choi have quantum properties that contribute to their extraordinary efficacy. For example, let's take zinc zetabiome. In zinc zetabiome, there is coordination of two fucose molecules, fucose is a kind of a sugar, to a zinc ion. The hydroxyl groups of the fucose molecules coordinate to the zinc ion through dative bonds. What are dative bonds? They are also known as coordinate covalent bonds. This is a type of covalent bond in which both of the shared electrons come from the same atom. This is in contrast to a regular covalent bond in which each atom contributes one electron to the bond. Dative bonds are typically formed when one atom has a lone pair of electrons and another atom has an empty electron orbital. The atom with the lone pair donates the electron pair to the atom with the empty orbital, and the two atoms share the electrons equally. Dative bond, that's the point, can exist in superposition states. A superposition state is a quantum mechanical state in which a system is in multiple states at the same time. For example, an electron can be in a superposition state of spin up or spin down, just as in the example of the two coins, head or tail. Superposition states of dative bonds are important for biological processes. For example, photosynthesis, one of the biological processes that is based on quantum entanglement, involves the transfer of electrons between different molecules. These electron transfers are often mediated by dative bonds in superposition states. Superposition states of dative bonds are also important for quantum computing. Quantum computers use superposition states to perform calculations that are impossible for classical computers. Therefore, the superposition states of the dative bond pertaining to the zinc-containing zinc zetabiome contribute to the quantum computations that are typical of our core, as well as to the quantum properties of the product. You do not find any other product in the world that has these quantum properties. Another interesting feature is that the products that I've designed and developed for Mr. Choi have also the potential ability to induce a time dilation at the level of human DNA. I was the first to describe this phenomenon in 2017 in this article published in the Journal of Neurology and Stroke entitled Alzheimer's DNA Vaccine and a Relativistic Time Dilation. Also, this article, like all the other articles I am mentioning in this talk, can be downloaded for free. They are in the public domain, so you can read them in detail. In this article, I described how interaction of a uniform, highly charged macromolecules, such as those characterizing both products I designed for Mr. Choi, interaction of these macromolecules with basic DNA binding proteins independently of DNA sequence. So this means that they can work on everybody. They introduce a type of gravity-induced time dilation that is not dependent on the individual genetic information. So everybody can benefit from those two products. Because of this, time will run slower for the DNA to which the macromolecules are bound. This will give extra time to the well-known DNA repair mechanism to perform their task and will slow down aging at the level of DNA. These highly charged macromolecules do not cross neither the plasma nor the nuclear membrane per se. So it's not so easy to compound a product with these properties. And this is precisely the reason why the other molecules of the two products that are so complex are introduced. This is, in a sense, the reconstitution of the primeval multimolecular structure that is responsible for the origin of life on Earth, that enables internalization of the highly charged macromolecules into the cells, ultimately reaching the basic proteins surrounding DNA. Slowing down the passing of time at the level of DNA will enable cells to repair mutations and or alterations of the epigenome, exploiting the repair mechanism that evolved over the course of millions of years, thus preventing the onset and development of almost all diseases. And finally, I wish to emphasize how the products that I have designed and developed for Mr. Hyuk Choi are based on original supramolecular assembly, as it is described in the article published in the Journal of Neurology and Stroke, and these exploit the principles of pleiotropy and redundancy. What is peculiar in the products is that each one of their components work on a separate, albeit converging, signaling pathway, and therefore fully implement the principles of pleiotropy and redundancy. What are these two words? Pleiotropy is the ability of a bioactive compound to exert many different types of responses. One compound, many responses, often on different cell types. Redundancy refers to the fact that many different bioactive compounds can induce similar signals. Pleiotropy and redundancy can be explained, respectively, by the ability of certain bioactive compounds to signal via more than one type of receptor complex and by the sharing of individual receptor component by more than one bioactive compound. In the products I have designed for Mr. Choi, pleiotropy and redundancy coexist in a perfectly balanced state that makes them absolutely unique. Because of the principles of pleiotropy and redundancy, the products I have designed for Mr. Choi show an overall activity that is much more than the sum of the activities of their individual components. These products are designed in such a manner that once released in the human body, their components self-assemble in a supramolecular structures that contribute to their enhanced efficacy as described in the article. The self-assembly is due to electrostatic and hydrophobic interactions that occur in the aqueous phase that in this case is represented by the gut environment in humans. The self-assembly is immediate upon release of the products from their vegetal capsule. Once the components are self-assembled in the structure described in the article, their biological activity is multiplied rather than added, because they can reach multiple targets at the same time and fully exploit the concepts of pleiotropy and redundancy. In other words, if the biological activity of each individual component of one of the products were 10, the overall activity of the self-assembled components would be 10 by 10 by 10 by 10, that is 10,000, rather than 10 plus 10 plus 10 plus 10, that equal 40. So 10,000 instead of 40. And also this is absolutely unique in the world and pertains only to these products. I wish to conclude this presentation by stating loud and clear my full commitment to support the endeavor of Mr. Choi, a very good friend of mine, to the fullest of my abilities. I am convinced that the collaboration with Mr. Choi is the most significant of my entire scientific career, and I feel that this represents the greatest honor and privilege. I thank you so much for your time and attention, and if you have any question, please do not hesitate to ask. I'm here to try to answer them. Thank you so much.